Hello everybody and welcome to a science video. Today I'll be doing a study video for my science final, which for me is tomorrow, and who knows when you're going to watch this video, so let's get going. So I'm using a PowerPoint that was created by my science teacher, Mr. Rossi, and it's about mitosis, and since I'm an honors bio, it obviously says honors bio. So there are different types of cells. There's somatic cells, which are body cells, and they have 46 chromosomes. And there are gametes, which are sex cells. They only have 20. They are egg or a sperm, and they're only composed of 23 chromosomes since they're supposed to come together and to create the first somatic cell of your body. Uh, there are limiting factors on how big a cell can grow. Uh, a cell size can vary greatly, especially depending on what task it needs to do. For example, red blood cells are 8 and nerve cells are 1 I don't know what the measurements are, but you get a pretty good idea on how different they can be. And cell size is limited. Diffusion, it decreases as a cell grows. It will take, because the bigger the cell, the harder it is to move things from one cell to the other cell, and it will take longer for waste to exit a cell or building blocks to enter a cell. So it'd take longer and longer to do things. So protein production is decreased. And although some people may think chromatin and chromosomes are interchangeable, they're not. Chromatin, it's DNA when the cell's not dividing. It's basically this over here. It's this big, jumbled, spaghetti-looking stuff. And so it's loosely packed and wraps around proteins, or histones. But what did happen there? But chromosomes are the DNA when the cell is dividing. It is these X's. And it's still a tangled mess, but at least it's in a format in which it can be split evenly. It's composed of tightly bound chromatin, and steps 1 through 3, DNA forms a chromosome, also known as a chromatid, and then the chromosome replicates. Duplicated chromosomes before division. So why do cells divide or are made? To replace old worn out cells. Think about it for those motorheads who may be watching. Slingshot, I'm talking to you. As worn out parts in a car. You have to replace them with new pieces. But since your body can't go and buy a new cell or a new organ, it has to start from scratch using stem cells or body cells to then replicate and make a new form. Or to replace injured cells. That's why a scab forms over your skin. That's basically blood that's clotted together to create a tight barrier so that the germs can be dealt with. And underneath that scab, new skin cells are developing to pretty much cover up the wound. And that's why scabs slowly go away and they don't just fall off. Or for an organism to grow. That's why children start out so small and get bigger. They have to build more and more cells. And their bones, which are actually in my right now, I am still considered adolescent and still growing, so my bones are composed of cartilage-like plates called growth plates, and these growth plates can actually grow larger and larger until they're too until they reach a time in which the hormones in my body shut off and tell them, okay, you're done building, now just keep everything together. Or for reproduction to occur. Wow, chicka wow wow. Yeah, I'm not going to go into depth about that one. And the cell cycle. Growth and division of somatic cells. Somatic cells and body cells are quite different, so they go through a slightly different process. A cell has two general periods. It has interphase, which is the growth period, and division, the production of two new daughter cells, which is basically what comes out from the original cell. But this division stage can be divided into more subcategories, into stages. Interphase, the three subphases. So the first part of interphase is G1, a period of rapid growth. S, or where it will begin, the restriction point is passed, and DNA is synthesized and chromosomes are copied. Or G2, all organelles are copied. This allows for both cells to have a full set of organelles. And it's also a rapid growth in protein, rapid amount of growth and protein synthesis. And the DNA is checked for error before mitosis begins. This is to make sure you don't somehow get, I don't know, a cell that can do something that it shouldn't, or has everything it needs. So as you can see, this is G0, which is a stage only, which is kind of a stage of like it's an odd stage. I'm not going to go into depth about it. So G2 it prepares for mitosis, which is prophase, metaphase, anaphase, telophase. Prophase. In prophase, the DNA coils and becomes chromosomes. That way they can split more easily. All chromosomes have already been duplicated and have two identical halves of themselves, called sister chromatids. See? Each individual of these things is a chromatid, and the whole thing is considered a chromosome. So when it splits, it is literally pretty much cut in half, and one side goes to one cell, one side goes to the other cell, and both of them have all the DNA that they need. And they're connected by a centromere, which is this little blue dot looking thing right here. And the nuclear envelope disappears, which is an envelope basically like kind of a membrane surrounding all the DNA. So you can see all these base pairs. This is the DNA strand. It then coils itself up a lot, and it kind of coils itself around these little histones, and then they become nucleosomes, and they get more and more of this stuff gets raveled and raveled tighter and tighter until you get this big X. And there are quite a few of these inside a cell. And during metaphase, the chromosomes, or sister chromatids, line up around the equator, which is kind of like how you would think it would be. They line up as if, just imagine giant X's are lining up on the equator of Earth. Microtubulars from the centrioles connect to the connector on the centromere. Microtubulars are basically, since animal cells do not have a cell wall, we have these centromeres that basically act as a network for transportation and basically hold the whole thing up. And during anaphase, all 46 sister chromatids split. One chromatid moves to each side of the cell, and each side of that cell will form a new nucleus for the new cell. See? So during this time, you actually have double the amount of DNA that you normally have in one cell. Until, during telophase, the nucleus begins to reappear, and chromosomes uncoil back into chromatin inside these new nucleus, or nuclei. Final step is cytokinesis. This is the actual breaking away of the two cells. You can see in this little animation here, it starts to get more and more peanut-shaped, and then just splits into two cells. This is not even technically considered of mitosis. The two cytoplasms pinch off and split into two. But instead for plant cells, they create a new cell wall between them and still stay partially connected. End result, two new identical cells. So here's a little flowchart. Pause the video if you want to go over this, but I will go on. Oop. And here is basically what it looks like under a microscope, or some sort of test. And another one of them. And that's the end of the video. I hope this helped, and have a good day. Bye!